Okay, so we've had a brief introduction to three lines of defense. We talked a little bit about inflammation, and we're going to go into more detail about some of the specific cells and some of the chemical mediators that are involved. So when we talk about the different types of immune system, you can look at the three different categories, the first, second, and third line of defense. You can look at whether it's specific or non-specific, but another way you can define it is if it's innate or adaptive. So here are two groups. I found this picture that's really handy from Nature Reviews, but you have the innate immunity, and this is the non-specific immunity. These are the cells that don't care what's in your body. They're just there to kill it. If it gets in there, their whole job is to try and eliminate it, no matter where it came from or what it is. The other group are called adaptive immunity cells. They adapt, they learn. These are the things that create long-term memory, like when you get vaccines and you remember that chemical for a long time or for your entire life potentially. So innate versus adaptive. And remember, when we're going through these cells, you want to make sure you notice some special features that make the cells stand out to you. What do they like to attack in particular, and what weapons they use to try and fight the invader? So it's kind of like learning about the military and the different branches and what kind of special tools they use. Some of them use similar tools. Like if you think the Air Force, you think planes and jets. But remember, the Navy, Top Gun, they're using jets too. So they have some of the similar weapons. And we'll do that. We'll talk about some of the chemicals that fight infections and how they're used by these different types of cells. So remember, innate versus adaptive is kind of the big two categories. So first let's talk about some cell mediators. And the innate response, remember, is anything that gets in your body that doesn't belong, your body will attack. And some of the keys to remember innate response is that it's non-specific. It doesn't care. It's immediate. It doesn't care what it is, so it's not going to stop and ask questions. It's like that old saying, pummel them now and then ask questions later, or shoot now and ask question later. That's what the innate system does. It attacks right away and doesn't care what it's attacking. It attacks stereotypically. So you kind of get this general response that happens every time, consistently, every single time from the innate immune system. It doesn't matter what the invader is, you get the same problem happening over and over. You get the redness, the swelling, the pain, a little bit of a fever. They're all the same no matter what it is. And then you don't learn. So this doesn't get better with time. It doesn't learn to be better. It's exactly the same now as it was 20 years ago in you. So it doesn't change. It may get worse eventually, but it doesn't get better. And then some of the weapons that it uses or detectors it uses are things called cell surface receptors. So these pattern recognition receptors are on the surface of lots of these innate cells. So something like a macrophage, they'll actually have receptors called toll-like receptors. And toll, this is kind of interesting, it's a German word for cool. So when they discovered that, hey, these receptors are kind of cool, so they called them toll receptors or neat receptors. So the toll receptors, these are like the most ancient immune components. These are very, very primitive, and they think it's one of the very first immune systems or immune structures that we actually had. So it's common across lots of different mammals and lots of different animals. It's very similar. The next one is going to be these complement receptors, and the complement receptors are the mannan binding components. These trigger a system called complement in your body, which we're going to talk extensively about in probably 6 or 7 slides. But complements is cascade that attacks structures in your body. And mannan is actually a chemical that you make that binds to these invaders. And the last one are scavenger receptors. And scavenger receptors are kind of important because they detect negative charges on the surface of most bacteria. So bacteria have this negative charge and it just knows, well, if this thing's negative, then we're just going to attack it. Unfortunately, down here with the LDL carries a slightly negative charge too. Low density lipoproteins. These are the bad cholesterols in your body. So what they think is happening is these scavenger receptors detect that and think it's foreign, and they cause things like inflammation. It causes you to get this infection-like response. And it blocks up the LDLs, it tries to attack them, it sticks them to the walls of the artery and makes things even worse. So those are the 3 pattern-like receptors. And the key with pattern recognition receptors is that most invaders, like bacterial invaders, have similar patterns on their surface. So they know what's going on. So here you can see this little toll-like receptor up on the surface. When it detects some kind of invader, it sets off this chain reaction. And the chain reaction is going to do things like try and kill the invader. So these patterns that are on the invaders are called PAMPs, so pathogen-associated molecular patterns. And bacteria are kind of like us. They evolve and they have special weapons, or they have special coats that have made them do really well. Our body recognizes these special weapons or special coats and then learns to attack it, and that's a PAMP. Right. Next, we're going to get familiar with the different cells. And something to keep in mind is that most of the cells of the immune system actually originate at one place, and that one place is in the bone marrow. They're blood cells. They're called blood cells. When you're talking about white blood cells or red blood cells, they're both blood cells, and they're both hematopoietic cells. They come from a blood cell, blood stem cell. So here are the different key components we're going to talk about. Mast cells, neutrophils, eosinophils, basophils. A lot of these you've already heard of, most likely. We're just going to talk about them in a little bit more detail. And then we'll talk about the macrophages, the natural killers, and the platelets. But you can see on that last slide how almost all of them come from this original stem cell, the hematopoietic stem cell. And then they can become red blood cells, white blood cells, platelets, etc. Right. So let's talk about the mast cells first. And the mast cells, basically there's big bags of granules. So you can see all these granules all over the place. And the granules are holding chemicals inside of them. And we're going to talk about these, they're really important. When you look at things like epithelium, so you've got this epithelial layer going on along here. Just under the epithelium, you have the connective tissue. And connective tissue is full of mast cells. So epithelium, like along your skin, if something penetrates the epithelium and actually makes contact with your mast cells, they degranulate. They release their granules. So these granules get dumped out. And that's the first thing they do. It's kind of like an octopus. You scare them, and their first line of defense is to spit out that ink. So that's what a mast cell does. You irritate it, you bother it, it degranulates and spits out these little bags of chemicals. One of the most important chemicals are called histamines. So the histamine comes out and it causes vascular reactions. It causes vasodilation and smooth muscle contraction. And I'll show you some pictures of that in the next slide. But you're familiar with histamine. It's usually associated with allergies. So where you find these mast cells are anywhere that somebody can penetrate the skin. So the skin lining on the outside, the digestive tract lining, the respiratory tract lining, you find lots of these mast cells. If it's irritating the respiratory tract and these degranulate, they start causing an allergic reaction basically, and they'll start causing your airway to close off. So that's histamine. It has a huge vascular and bronchial effects too. And then other factors, these are called chemotactic factors. Remember that chemotaxis? These chemicals attract other white blood cells. Like this, neutrophil chemotactic factor. Guess what cell it attracts? It attracts a neutrophil. So this neutrophil has a high affinity to this neutrophil chemotactic factor. When it's released, the neutrophils come rushing into the scene right away. Neutrophils are actually going to be the first on the scene of an infection. This is part of the reason why. Another chemical is eosinophil chemotactic factor. This is attracting eosinophils, and eosinophils primarily are there to attack parasites or come out in allergic reactions. And we'll talk about them in better detail too. So if you get this big parasite that's invading you want the eosinophils on the scene right away to try and stop it. That'd be like a huge monster getting through your house. You don't just wait for it to destroy everything. You try and get in there and kill it really, really fast. So that's the first thing that happens. All these granules are released. These granules are pre-made. They're just stored up. And when you scare the mast cell, whether it's pressure or chemical agents or like a bacteria or anything that irritates this, it degranulates all over the place. Once it's degranulated, its weapons are all dumped out. Now it's going to start making a new set of weapons. So this new set of weapons is through a synthesis process. And that's on the next slide. I think it's on the next slide. Or, sorry, it's the slide after that. So here's an example when you degranulate. There are the different chemicals that are released. So histamine is dumped out. There's the mast cell dumping the histamine. Histamine goes over to the blood vessels, causes smooth muscles to constrict, but the capillaries to retract. So the smooth muscles will squeeze down a little bit to slow blood flow, but at the same time, the capillaries open up to allow fluids, exudates, transudates to escape out. So histamine is kind of important. Well, I already told you what it does to blood vessels. It causes smooth muscle constriction and capillary dilation. But parietal cells is the stomach mucosa. If histamine is released in the stomach or gets exposed to the stomach, it actually releases acids. Why would histamine want to release stomach acids in the stomach? Think about it. If an infective agent got into your stomach, It's penetrating through the wall and it stimulates a mast cell. The mast cell releases histamine. Histamine goes to the parietal cells and dumps acid, hydrochloric acid, all over this invader. So it's trying to kill the invader. So sometimes if you just take a step back and look at it, they're kind of self-explanatory. And then the synthesis mediators. So once it's degranulated, then it starts synthesizing these other products. And here are the three main products. Leukotrienes. Leukotrienes are, it's basically saying that white blood cells, leukocytes, talking to each other. So these are chemicals that talk from one leukocyte to another leukocyte, right? They can also have some vascular effects too. Prostaglandins. So we've talked about prostaglandins a little in the past. We're going to talk about them again. But prostaglandins have a tendency to do things like sensitize the body to pain. But they also have similar effects to leukotrienes, where they can cause vasoactive effects. Right? They originally named the prostaglandins because of the gland that they found it in, the prostate gland. Yeah, so prostate gland actually helps activate semen. That's one of the factors, but it's also released in other parts of the body too. And then platelet activating factor, you can guess what that does. It activates platelets. So it's activating the platelets to try and clump up or block the blood flow. So if something cut through the skin, it's going to try and activate platelets in that area to stop any bleeding from that area. Okay, so one of the disorders that could happen is you can actually have something called mast cell activation disease. And the one that's most common is called mastocytosis. And I know you can't see my air quotes. I'm actually doing air quotes here and I'm laughing at myself, but mastocytosis, when I said it's more common, is they actually thought it was a really rare disease, but what they're finding is that they think maybe it's more common than they thought it was. Because we're looking at all of these disorders that have very vague symptoms that kind of overlap with the symptoms of mast cell activation or massive mast cell activation. So the mast cells have a tendency to open up blood vessels, they have a tendency to constrict airways, they irritate GI tract, they affect blood vessels. So when you have too many mast cells that are overactive, you're going to see effects in the GI tract. So they're thinking that some types of irritable bowel syndrome may actually be a form of mastocytosis. Too many mast cells releasing too much histamine, causing too much irritation, redness, swelling to the GI tract lining. They're thinking that with fibromyalgia, it may be affecting pain signals, too much histamine. And then they're also looking at airways, so different forms of anaphylaxis and with bloodways, anaphylactic shock may actually be related to too many mast cells. And then, of course, the chemical you want to keep in mind, histamine. All right, next, the leukocytes. So the leukocytes, you've seen this before, you've gone through this before. We can measure the leukocytes by just taking a little blood smear. So we can take a blood sample, we can spin it down, we can smear a little bit on a slide, and we can look at it. But here are the primary players when we talk about white blood cells or leukocytes. So the neutrophils are going to be the most abundant, and they're also the first on the scene. Down here you can see the percentages of each of them. But they're the most abundant in the system. They're the first on the scene of any invasion. So they get in there within the first 24 hours and just start trying to kill everything. So they like to eat their food. They eat their prey, and they spit out enzymes on them to digest it. I have a slide for this, so I guess I don't have to speak too much about it. But like I said, we can take and put a little sample on a slide and look. We can count the red blood cells versus the white blood cells on the slide to see if you have too many white blood cells or not enough. We can look at different types by looking at their nucleus, looking at the color of the granules inside of them. Do they have granules? Do they have a large nucleus? So we can describe them. We can spin down the sample of blood and take a hematocrit. We can look at the plasma, the red blood cells, and this little buffy coat that's white blood cells and platelets. And then you can see, like we've seen before in leukemia when we talked about different forms of leukemia, that buffy coat gets thicker because you're making lots of white blood cells. So normally this is less than 1% of all the spun down blood, but you can see in things like leukemia, you can have high percentages, 20 or 30% of your spun out blood can actually be that buffy coat. So let's talk about each of those one at a time. The first one's a neutrophil. So neutrophils are also known as polymorphonucleocytes, and PMN is what you usually see. So it stands for polymorphonucleocytes. So poly meaning many, morpho meaning shape, and nucleocytes talking about the nucleus of the cell. When you look at this nucleus, they usually have these multi-lobed or strange lobed nucleus in them like these. Right? So the keys you want to remember is that they're the first on the infection, they're the most abundant, and when you see disorders that are associated with it, if it's too low, we call it neutropenia. So if you don't have enough white blood cells, specifically neutrophils, this type's called neutropenia. When you see this, usually it's because they have overwhelming infection. So they have an infection somewhere, and all those neutrophils are going to the infection. So if you take a sample of blood, they're really low in neutrophils. You can have autoimmune disorders or reactions to drugs like steroids can actually cause a change in neutrophil count in the blood. And then you're going to see kind of common themes like bone marrow damage. Where are these things made? They're made in the bone marrow. If you have massive bone marrow damage, would you expect just the neutrophils to be low, though? Probably not. So this is something right here, the overwhelming infection kind of pinpoints at neutrophils. And over here, high neutrophils, it's called neutrophilia. And in this situation, acute bacterial infection, so a new infection, a fresh infection, your body's going to crank out lots of new neutrophils, and they're going to come rushing into the blood. So you're going to see a raise in the neutrophils. And I'm kind of pointing these out because when you look at a spun down sample of blood and they do a differential, so they see that buffy coat's pretty thick and then they spread it out onto a slide, they can look at the percentage of the white blood cells, and if they see high neutrophils, then they're probably going to suspect you have a fresh infection or a new infection. So if you were walking into a doctor's office, one of the first things they want to do is, act like a vampire and take a sample of your blood. But if they see high neutrophils and they go, you have an infection somewhere, we need to, give you an antibiotic or whatever to try and fight the infection. Okay? So neutrophils are usually the first on the scene, especially they like to fight bacteria and fungus. So those two things travel a lot, and they're trying to kill them off or stop them. Next, eosinophils, or eosinophils, if you want to be really specific with the pronunciation. But eosinophils, they stain red is how we identify them. So they have a bunch of granules inside of them to help break down whatever they're trying to attack, and those granules stain red. Right? So normally eosinophils or eosinophils are really low in numbers. So having a low blood sample of these usually isn't really indicative of anything because you're always going to be a little low in it. It's when you're high that you have to be suspicious. So eosinophilia is when you have high eosinophils in the blood. And usually high eosinophils is a sign of a parasitic or an allergic reaction. So parasitics and allergics, because their main purpose is to attack parasites and come out with allergies are present. So if you spin down a sample of blood, you can see there's the buffy coat. If you looked at the buffy coat in somebody that has a parasite, like malaria here, you can actually see the buffy coat is going to be a little bit thicker. You can break it down and look at the platelets and the white blood cells. You can see some of these quote-unquote eggs, so immature asexual forms of the parasite, and then you can see some of the adult forms of the parasite down here by the red blood cells. So remember, the malaria worm loves red blood cells, and when you spin it down, it's a large organism. It's going to be heavy, so it's going to spin down here with the red blood cells. So here's just an example of what might happen, right? And then other situations like leukemias. Leukemias are overly producing white blood cells. So if they're overly producing white blood cells, you may just see higher eosinophils. All right, and then the basophils. The way I always remember basophils are basically just circulating mast cells. They're just like a mast cell. They're full of chemicals called histamine. They have chemicals called heparin as an anticoagulant. So they work almost exactly the same as a mast cell, except they're circulating in the blood instead of being stuck in connective tissue. So here's an example of a resting mast cell. You can see it. Here's one that's degranulated. Here's another example, one that's degranulated. You can see all the broken edge of the membrane there. So these are partially degranulated. So normally, just like the eosinophils, you're going to have low basophils. So not usually is it significant if you have a low reading. But when you have a high reading, it usually tells you what's going on. You walk into the doctor, you're feeling sick, you're feeling weak, you runny nose, you don't know what's going on. They take a blood sample, it comes back, and then, ta-da, high basophils. The first thing they want to say is you probably have allergic reactions. You're allergic to something. Maybe it's a season. Maybe it's something you ate. Right? Usually it comes in common with inflammation too. So remember the histamine causes swelling. And then some leukemias will do that too. All right. Next ones are the monocytes and macrophages. And I put on here monocytes and macrophages, but technically a monocyte is what's circulating through your blood. It's always circulating around and around and around. But when it's activated by one of those cytokines, then it's turned into a macrophage. It's like Clark Kent over here, the monocyte with his little kidney shape or horseshoe shaped nucleus. And then you activate it, and it runs over to the edge of the blood vessel, like its telephone booth, and then turns on its super suit and becomes super macrophage. And it's out there to start eating away at things. On the next slide, I have a homework question. Macrophage are actually found all over the body, and we give them different names. A monocyte is what we call it when it's floating through the blood, basically in an inactive form. When it's activated and moves into tissues, we call it a macrophage. So I kind of actually already answered this. What we call a macrophage when it's in the bloodstream, it's called a monocyte. But what are they called, and you can Google this, you can look it up in your book, what are they called when they're in the lungs? What are they called when they're in the liver, or the brain, or the spleen, or the kidney? These are terms you want to get familiar with, because when you see these names later, automatically you should think macrophage. Macrophage have a specific type or pattern of behaviors. They're big eaters. They're to eat other cells. So if you recognize these names, like the one in the liver is a really common name, or actually I know if you took physiology with me, I really stress the one that's in the brain, but when you see these, you know exactly what they're going to do because you understand a macrophage. Right? And then where are they all made, and then how long do they typically live on average? Right? And you'll find there are patterns. You don't have to necessarily go through and look at every single one. You can find patterns to it. Right? So macrophage's main job is phagocytosis. We already started talking about this a little bit. We talked about the process. When you activate that macrophage, it starts sliding through here. Remember that margination, or it's also known as pavementing. So you're pavementing along, and I love that because it goes perfect with my 4x4 example. So you hear those big tires going running along the pavement. And then they merge down into the cells, into the tissues, through diapedesis. And they're moving, remember, what's driving them? What C word? What process is driving them? It's chemotaxis. So they're driven by chemotaxis. They're detecting chemical messengers in the environment here. And they're getting closer and closer to the invader. And they're moving in. And once they find the invader, they're going to go up to it, just like this one. It's going to wrap itself around it, engulfs it. So it doesn't actually break the membrane. It doesn't break the membrane of the macrophage. The macrophage actually wraps its membrane around the bacteria and then just pinches it off and swallows it whole. That would be like you taking a hamburger, wrapping your mouth all the way around the hamburger, and without chewing, just gulp, swallowing it. The hamburger didn't penetrate any membranes. Now it's encapsulated in your stomach and it's being digested. Right? So let's look at that process of digesting the bacteria a little bit closer. So you have a couple steps. The macrophage comes along the bacteria and it adheres. So it glues itself to it. Grabs the bacteria, so it's stuck to the surface of the macrophage now. Now the macrophage is going to engulf it, wraps around it. Once it's engulfed it, now it's isolated. There's your bacteria isolated. These little things are lysosomes. So the lysosomes come floating over. And I shouldn't say float, they're actually transported across the cytoskeleton to meet up with this phagosome. So phagosome is where the bacteria is at, the spot that it ate. The lysosome contains those hydrolytic enzymes. When they meet up, you actually have a phagolysosome. So now it's like you spitting or dumping your stomach on this bacteria. The bacteria is digested, broken apart, it's cleaned up. Your cells, your macrophage is going to swallow some of those amino acids, maybe swallow some of the good lipids, and it's going to spit the rest out. So just like you, eating a sandwich goes through, you digest it, you take what you need from it, and you defecate the rest out. It's the same process. Right? So I want you to look kind of at how a phagocytosis can actually promote inflammation. We're going to talk about that on the next slide. Or actually, not this slide, but the next slide after it. So here's just another picture I found that I thought was really cool, and it explains the whole process again. So here you have enzymes that are made at the rough endoplasmic reticulum, move to the Golgi apparatus, packaged in vesicles, so the lysosomes. Lysosomes merge up with this phagosome right there, and then you have a phagolysosome. You digest the particles, absorb what you need, and then exocytose the rest. So I think macrophage are actually really cool. So macrophage, how they contribute to inflammation? Because they have specialized chemicals in them. So they have these interleukins. Interleukin means between white blood cells. They communicate between white blood cells. So interleukins are usually abbreviated with the letters IL, like IL-1, and I think if I remember right, there are over 23 of them. So there's IL-1, IL-10 is another common one, IL-6 is another common one, but there are lots of these interleukins that talk between. It's like a special language between the white blood cells. So it also makes something called tumor necrosis factor. What they found is when they first discovered this tumor necrosis factor is that it helped tumors to go through apoptosis. or I shouldn't actually say apoptosis, it's necrosis, help these tumor cells to blow up or burst or die. And they thought at first, well, geez, look at that, we found the cure to cancer, but it's a little bit more sensitive than that. So they call it a tumor necrosis factor, but it has a lot of effects. Here you can see the general effects of some of the interleukins and tumor necrosis factors. So acute fate-resistant reactions, causing fever, making you want to sleep more, decreasing your appetite. So all those symptoms you have when you have an early infection. They stimulate neutrophilia, which remember is increasing the number of neutrophils in your blood. So endothelial effects, the effects on the blood vessel. You can see that it allows white blood cells to stick a little bit more. So these macrophages, when they release these, they're telling their neighbors or other white blood cells, hey, come on in, the party's this way. It's stimulating these endothelial retraction factors to help open up the endothelial cells a little bit. They turn on fibroblasts to come in and start fixing or healing the tissue. And then leukocyte effects, they attract other leukocytes, white blood cells. So you can see an overall response of the macrophage. Right? And then the macrophage will also make things called ROS. Do you remember what that means? It means reactive oxygen species, or free radicals. Right? So it'll make these things to help digest or kill the invader. It also makes nitric oxide, which has a couple effects. One is that it causes blood vessel dilation, but nitric oxide is also considered a free radical, so it can help break down the invaders. So let's say that you have some kind of invader like a virus or whatever, and you engulf it, you eat it up. This macrophage can spit those reactive oxygen species on there and try and kill and destroy that virus or bacteria. So remember, monocytes, they're basically the precursor to macrophage. So low monocytes, usually if you have one low count, they don't really get worried about it, but when you have consistent low monocyte counts, then they get a little bit freaked out at the doctor's office. So high, if they're way high, it could be because you have a chronic infection. Chronic infection means that it lasts for, well, for the monocytes to start popping out at least 3 days, but chronic is usually referred to as over 2 weeks of an infection. So things like tuberculosis, fungus infections, those have a tendency to overstimulate monocytes to replicate, so you usually see them higher in the blood. Bacterial endocarditis, so leukemia that focuses on monocytes, so monocytic leukemia or myelomonocytic leukemia. So you see a lot more. But the problem is with the leukemias is they make lots of white blood cells. Unfortunately, these white blood cells, they don't work so well. So yay, you have more white blood cells, but what's the point if they don't work? It's like having a collection of cars and none of them have engines in them. It's kind of pointless. Right. Oh, I was going to talk about the pictures here. So here you see the skin. Lots of monocytes getting in here and actually causing damage to the skin. This is interesting because when the monocytes, when you have the leukemia, what they can actually do is they get kind of hungry, so they'll eat your red blood cells, and it's called erythrophagocytosis. So this sometimes happens when you have acute myeloid leukemia, which I had you look up for a homework question on the last set of videos. But when you have way too many of these white blood cells, especially these monocytes, they're phagocytes. I mean, they're hungry, so sometimes they have a tendency to eat the red blood cells around them. It's called erythrophagocytosis. So erythro meaning red blood cell, phago meaning eating, cytosis is cells doing it.